We're just the kids in school. Each day we learn some more. We love biology. We're Kapp kids. So many things to learn. Each day learn something new. We'll share them with you. We're Kapp kids. My grade is turning, changing each day. With Miss Kapp as my teacher, I'm finding my way. Learning bio's not so tough, except when we've had enough. But there's lots of fun stuff. We're Kapp kids, Kapp kids, Kapp kids. We're Kapp kids. That's us.
Hey kids,
We're the Kapp kids.
Don't you wonder why each of us looks so different? To answer, is it DNA and proteins? To really understand though, we have to learn about what DNA is and how we can make protein in our body.
First of all, DNA is like the recipe to build a cake, and we are that cake. DNA instructions are very different in every human body, just like how there are different recipes for different cakes, like chocolate, vanilla, and strawberry. Protein is the building block for our bodies.
DNA is like the nucleus of our cells. DNA is shaped like a twisted ladder, which we call a double helix. It is made of repeating chains of nucleotides, which are the little pieces that make up DNA. In a nucleotide, there are sugars and phosphate groups, which are connected and make the side strands of the DNA. And then there are nitrogen bases, which make up steps of our DNA ladder.
On each side, one base connects to the other. The four nitrogen bases are adenine, thymine, cytosine, and guanine. Adenine and thymine connect together, while cytosine and guanine connect together. Imagine that adenine and thymine are puzzle pieces that fit together, while cytosine and guanine are the other ones that fit together separately. Cytosine and thymine can't connect together, only thymine and adenine.
Similar to DNA is RNA. RNA is like a copy of DNA, but while DNA is a twisted ladder, RNA only has one strand, similar to a spaghetti noodle. RNA is pretty similar to DNA still, as it still contains four nitrogenous bases. But while DNA has bases adenine, thymine, cytosine, and guanine, RNA has the nitrogenous base called uracil in replacement of thymine. Remember how the nucleotides of DNA have sugars? DNA has deoxyribose sugars in its nucleotides, while RNA nucleotides have ribose sugars instead. Finally, DNA is only found in the nucleus of the cell, while RNA leaves the nucleus into the ribosomes of the cell.
Now that we know the differences between DNA and RNA, let's start talking about how they make protein. In our cells, there's a process called protein synthesis that occurs. This is like how construction workers build a house. The main blueprints of the house are in the contractor's office. The construction workers copy parts of the blueprints to make their specific parts of the house, like the walls, or the windows, or the roof.
DNA is like the main blueprint. It contains the information and instructions for the building of protein. mRNA, or messenger DNA, is a type of RNA that copies specific parts of the DNA and sends it out of the nucleus to build the protein. This is like the construction workers that copy the parts of the blueprint.
Now, the first part of protein synthesis is called transcription. This is the part where the RNA copies a section of the DNA. In the nucleus, the DNA unwinds, no longer being a twisty ladder. Then, an enzyme unzips the DNA like a zipper, breaking the bonds between the nitrogenous bases. Then, the mRNA nucleotides connect to the unzipped section of DNA, with the bases matching up.
Fun fact, thymine in DNA and uracil in RNA have super similar structures, which means that they can serve the same function. Codons are used in our DNA sequences as code. Codons are found in groups of 3, often referred to as triplets. Take a look at the strand of DNA. Can you match the unzipped DNA strand with the proper RNA nucleotides? Good job. The G goes with the C, the A goes with the U, the C goes with the G goes with the C, the A goes with the U, the T goes with the A goes with the U, and the C goes with the G. An easy way to remember this is apple under the tree and car in the garage. A to U, C to G. Once the unzipped section of DNA has been copied, the mRNA detaches from the DNA, leaving it as a single-stranded mRNA. This is our mRNA strand. This is the step of protein synthesis called transcription. After this, the mRNA leaves the nucleus like the copies of the blueprint. The mRNA goes to the cytoplasm, into the ribosomes.
The next step to protein synthesis is translation. This is the process that's used to bake a cake. Because our recipe is kept secret, mRNA has to make a copy of the recipe, DNA. The mRNA we call it our messenger owl, similar to Harry Potter owl named Hedwig. This is due to the fact that DNA can leave the nucleus, while mRNA can go into different parts of the cells. The ribosomes, once the mRNA reaches the ribosomes, it waits for the tRNA to arrive and piece together.
Let's wait for the tRNA to arrive. The tRNA carries the amino acids to piece them together to make the protein, based on the instructions in the mRNA. This is like Legos. Do you think you can make Legos without the instructions? That would be very hard. Luckily, the bases of the mRNA are split into triplets, grouped into 3 bases each. Each triplet is a code that fits with a specific amino acid, one of 20. Here is the codon chart that tells us which triplet matches with which amino acid.
The overall shape of tRNA is similar to a pitchfork. The tRNA contains these anticodons at the bottom of their shape, which have 3 bases that correspond and match with the codon on the mRNA. Look, this codon AUG fits with this anticodon UAC. The tRNA also has a specific amino acid that it codes for at the top. This one has methionine. The ribosomes are used to complete our building of our protein, which is the cake.
Let's work to solve this code. First, look at the codon. Second, use the chart to find which amino acid fits with it. Let's do the first one together. The first codon is AUG. So we look at the codon chart, we follow the path. First letter is A, second letter is U, and the third letter is G. Third, the amino acids, methylene. Can you follow the steps for the remaining codons? The answer is methylene, adenine, guanidine, phenylalanine, glutamic acid, and phenylalanine, and stop codon. As you can see, the tRNA matches with the corresponding mRNA codons that code for specific amino acids. Then the tRNA bonds with the amino acids to the chain. Then the tRNA detaches itself from the mRNA and leaves behind the amino acids. Once the chain is done, we have made our proteins. Yay!
Overall, DNA is like a recipe to build a protein. These proteins are what build our body.
However, the DNA can't leave the nucleus to get to the ribosomes, the build sites, which is why we have mRNA.
mRNA is like our messenger owl that copies the instructions of building protein and sends them to the build site, or ribosomes.
The ribosomes reads the secret instructions or mRNA and builds our proteins or cake.
Hooray!